Hi everyone. So today I'm talking with Christian Setz. Hi Christian.
Hello.
So Christian is the CEO of Immunologik, a startup based in Berlin that is developing a new type of drug against HIV. So although today most HIV infections can be kept under control with antiretroviral therapy, in some people the HIV virus can become resistant to these therapies, which means that thousands of people still die each year. And that's the problem that Christian is trying to solve in particular. So Christian, can you tell us a bit more about how your drug candidates can fight HIV and how it is different from what's currently used in the clinic?
Yes. Our drug, EML or 106, attacks cellular proteins in contrast to the so-called combined antiretroviral therapy, which is at the moment the standard therapy for HIV infected people. And our drug attacks a specific cellular proteins which are crucial for the replication of HIV-1. And we identified one special protein, and this protein belongs to a group of cellular proteins called deubiquitinating enzymes. We call it short DUBs. And you have to know that HIV has no own metabolism. So HIV is depends on the replication of HIV depends on the function of cellular proteins. And when you block these cellular proteins, HIV can't replicate anymore. So this is complete contrast to the standard therapy. The standard therapy attacks viral proteins and blocks viral proteins and causes a defect in replication. So that's the biggest contrast to the standard therapy.
Okay, I see. And so, in terms of the development of this drug, how far are you? What kind of results you have to test if it actually can work better than current therapy?
At the moment, we finished the preclinical studies. So we identified the cellular target and established a model, a preclinical model, to study the influence of our drug EML 106 in an ex vivo model. So there are no good animal models for the HIV infection because HIV infects exclusively human cells and not cells from mouse or apes or something like that. So we had to establish a preclinical model, and we used tonsils from standard tonsillectomies. We got these tonsils from the clinic, and then we prepared these tonsils. And these tonsils contains all relevant cells which are necessary for an HIV infection. So we got T cells, we got macrophages, dendritic cells, and so on. So these tonsils are a very good model for that. And then we can infect these cells with HIV and apply our IML 106 and study the replication, the influence on the replication of HIV, of these HIV infected cells. And we did different treatment options. On the one hand, we did a permanent treatment where we apply our drug during the whole time we observe the replication of HIV-1. It was 50 days in our case. But we did also a structured treatment where we only treated the cells on the first and the third day after infection. And in both cases, we could detect a dose-dependent reduction in replication capacity in this preclinical model. And of course, we also tested if there were any toxic effects on the cells. And we, in the effective range where the drug blocks the HIV-1 replication, we got no toxic effects.
Okay. Yeah,
So
I feel like there's something interesting about what you're saying. So I guess like when you're trying to develop something, you get some unexpected challenges, maybe like not having an animal model. So tell me a bit more on about like how you discovered the therapy, why you decided like to make a startup, and what sort of challenges you are encountering. Yeah, trying to move it into an industry instead of staying in academia.
we started our research on the virology in Erlangen. That's an institute which belongs to the university clinic in Erlangen. And first of all, we did a year-long basic research, of course, to identify this cellular target, because nothing was known before about the role of this deubiquitinating enzymes in the viral application. So this was the first step. And then a few years ago, first inhibitors of this deubiquitinating enzymes become available. And this gives us a good chance to study if these enzymes were really crucial for the virus replication. So this was the start. And then we got this very promising results. And we decided that it may be possible to develop an antiviral drug which targets these enzymes. So this was the start of this whole project. Basic research. It's always first basic research, then you can do an development.
Yeah, and how is the experience so far? Are you finding it challenging?
Sorry, I got the last question.
I'm just wondering, like, how are you finding the experience of working in a startup so far? Is it challenging? Is it exciting?
Yes, it's very exciting, and it's a very interesting thing, because you not always have the chance to see something in the laboratory, and then to have a chance to develop this. So it's very interesting, this whole thing, and let's see what happens in the future. But not every scientist has a chance to make these things, to bring an idea in the practice. That's interesting.
Definitely. And so now talking a bit more broader about HIV. So there's obviously like a lot of people trying to develop drugs and solutions. But what would you say, like from your own experience, are the biggest challenges to make a therapy that actually works?
Yes. First of all, it would be good if we have the chance to reduce the risk of resistance. That's one of a big challenge. And we have here the possibility, by blocking cellular proteins, which have a million times lower risk to get resistances as when you block viral proteins. And that's an important thing nowadays, because the most drugs which are on the market targets viral proteins. The second thing is that that we have a reduced toxicity, because I mean, the drugs of the standard therapy are well tolerated. And when I compare these drugs with the drugs in the 90s, there are there are not that much side effects. However, there are side effects, and especially the long-term side effects. No one can say if this side what are the consequences of these long-term side effects. For example, on the liver or on the renal, there are no reliable studies on that at the moment. And that's another big challenge, because some HIV infected patients has to stop the standard therapy because they have intolerability to the current drugs. So that's another challenge. And of course, the medical adherence is very important. So when you look in the 90s, in the mid 90s, an HIV patient has to take over 20 pills a day. So that's a huge amount of pills. And when you forget one of these pills, maybe the whole therapy doesn't work. So it's desirable to have an therapy with only one pill. So that's easier for the patient, and you have, and it's more reliable than when you take 20 pills a day. And of course, it's another challenge is to have a high efficiency of your drug, to really get an block of virus replication. That's another important thing when you develop a new drug. So these are the major challenges today.
So, yeah, actually, it's interesting. Obviously, you're still in an early phase, but you were mentioning all these challenges and things that need to be addressed. So, about the drug in particular that you're developing, are you intending it as an addition to current therapy, like antiretroviral therapy to avoid resistance, or do you see it becoming like a therapy that doesn't need all these other pills on top of it?
Yeah, I mean, I have to say we don't intend to replace the current therapy, because the current therapy works in most patients. However, there are about 80,000 people only in USA and Western Europe which develops a resistance against all available classes of the standard therapy. And then these people have a high risk very high risk to develop AIDS. And when you have developed AIDS, you have you die on the consequences of this immune disease. And yeah, we think that we want to help, especially those people where the standard therapy fails, and that's about 80,000 only in USA and Western Europe. And I mean, when you have to stop the standard therapy, I think it might be possible. We have to check this out. That our drug can be as a monotherapy, but it's not our intention to replace the standard therapy.
Okay. So the idea would be that when someone gets diagnosed, he takes the normal therapy, and on top of that, your therapy.
Yes, when they develop resistances or intolerance against this therapy, we will help this. It's our intention to help these people.
Okay, yeah, definitely. And so, is it intended as like a chronic treatment that you have to take for life, as it happens with antiretroviral therapy?
Yeah, I mean, everyone has to know when you got an infection with HIV, this virus persists alive for the rest of your life in your body. So you have to take every day your medications to block the viral replication and to control this disease. And the CART, so the standard therapy, is a chronic treatment, and our therapy will also be in chronic treatment. So the people have to take it for the rest of their life. But I mean, it's important to keep this in mind, that the virus never disappears from your body. So it remains in your genome, and you have to control this infection for your life, for the rest of your life.
It's interesting that you say that, obviously, Because so we hear about a lot of companies lately trying to use immunotherapies to treat HIV. So, and we hear from some of them that they claim that potentially, like basically an immunotherapy that uses the immune system to attack HIV. Like some people say it could be a cure for HIV. So, do you think that is true? Do you think that is possible, or are you skeptical?
I mean, it's very hard to get a cure from HIV, because it was shown that when you try to cure HIV, you have to kill all infected cells. When only 20 to 40 cells remains in your body, it's not that's nothing. 20 to 40 cells. And you stop the antiretroviral therapy, HIV come back in your body and replicates again. So you have again to applicate the standard therapy. So that's a really big challenge to eliminate all infected cells of your body. And the problem is that HIV is inserted in your genome and remains inserted in your genome. And there are, for example, long-lived T cells. And in this long-lived T cells, HIV can persist as a reservoir. That's always the questions of the HIV-1 reservoir. And your immune system can kill these cells, because HIV is hidden in these cells. It doesn't replicate for a long time. It can stop the replication for years, for example. So the cells survive, but HIV also survive in these cells. The genome of HIV remains in these cells, and at any time point, HIV can replicate again, and then you have again an efficient replication in your body. And if 20 cells survive, this can again infect millions of other cells. So that's a big challenge. But of course, it's interesting that a lot of companies try to cure this infection. But today, I think no one can give a reliable forecast if a cure would be possible one day. But I mean, one of the most frequent approach today is the so-called shock and kill strategy. Yeah. A lot of companies try to develop drugs which are used in this shock and kill strategy. And shock and kill, what means this? First of all, one drug, so-called latency reversing agents, try to get an expression of HIV in these latent infected cells. So that's the first aspect of the therapy. And the second aspect, the kill strategy, is to have an efficient immune response, so that your immune system can then kill this latent infected cell. So first of all, you have to shock the cell so that HIV is expressed again in this cell and is not hidden anymore. And then an immunotherapy should kill the cells. But as I said, if only 20 cells survive, you have a remission of HIV. And of course, this whole therapy must be given together with the antiretroviral therapy.
Okay, so more.
Yeah. And another problem with this therapy is that there are a lot of side effects. As you can imagine, not only the genome of the HIV is expressed in a higher level, but also of cellular proteins. So there are, for example, inflammations when you get this therapy, or fever, or also neurological diseases. So it's very challenging to develop such an therapy today. And I cannot say if it would possible one day, but at the moment, I don't think that a complete cure would be possible. But it's important to make this approach, of course.
Yeah, of course. I mean, I guess it's important to be realistic so that we can develop a real solution for the people.
I mean, it would be a sensation if this would be possible one day, but I think in the next 10 to 20 years, I see no chance for a complete cure of HIV. So it's important to develop also other options for the patients.
Yeah, definitely. Okay, then I think that's it for today. Thanks a lot, Christian, for explaining all these things. And
yes,
all the best with your company and your drug development.
Thank you. Bye.
Bye.